Students, today's topic is photochemistry of vision. I hope you have gone through the previous topics related to eye, namely the anatomy of eye and the optics of vision. Next is the physiology of vision. Physiology of vision is a complex phenomena. The main mechanisms concerned with vision are initiation of vision, that is phototransduction, which is a function of photoreceptors, that is rods and cones. Second, processing and transmission of visual sensation, which is a function of the image processing cells of retina and visual pathway. Third, visual perceptions, a function of visual cortex and related areas of cerebral cortex. Photochemistry of vision is the first part of the physiology of vision, which deals with the chemical changes in the retina, which enables us to see. Before going to the photochemistry of vision, let's have a quick recap of the anatomy of the eye. As you know, light passes through the lens system and the vitreous humor to enter the retina. Retina is a thin transparent membrane which exhibits three distinct areas: optic disc, macula lutea, and peripheral retina. The light passes through the cornea, aqueous humor, through the lens, vitreous humor, and finally it falls on the retina. Behind the retina is the choroid, and this is the sclera part. The retina ends as the serrated anterior margin of this retina is the ora serrata, and this is the optic nerve, optic disc. This is optic disc through which the optic nerve passes, and this is the fovea. So here you can see this is the picture through a ophthalmoscope. This is the picture of the fundus of eye through an ophthalmoscope. You can see here, this is the optic disc. It is a well-defined circular pink color disc of 1.5 mm diameter. It has only nerve fiber layer, so it does not excite any visual response. It produces blind spot in the field of vision. This is the macula lutea. It is a comparatively dark area situated at the posterior pole temporal to the optic disc. Its central depressed area 1.5 mm in diameter is called the fovea. This is the fovea. Central part of the macula is known as the fovea. Fovea or fovea centralis, which is the most sensitive part of the retina. The visual acuity is maximum in this part of the retina. Next, just have a recap of the layers of the retina. From outside to inside, the layers are pigmented layer containing single layer of cells having melanin pigments, layer of rods and cones, then external limiting membrane, outer nuclear layer containing the nuclei of the rods and cones, the outer plexiform layer containing presynaptic and postsynaptic elements of synapses that exist between the photoreceptors, bipolar cells, and horizontal cells. Next is the inner nuclear layer containing the cell bodies and nuclei, bipolar cells, amacrine cells, and horizontal cells. The seventh layer is the inner plexiform layer. It is the layer of synapse between bipolar cells, ganglion cells, and amacrine cells. Eighth layer is the ganglionic layer consisting of ganglion cells, which are the output cells of the retina. They transmit visual information to the brain. Next layer is a layer of optic nerve fibers consisting of the axons of the ganglion cells which form the optic nerve. And finally, the inner limiting membrane. It separates the retina from vitreous. In some of the books, also in Guyton, you will see that there are only 9 layers of the retina. That is this external limiting membrane. This has not been included as the layer of the retina. So it is 9. So either in some of the books it is written as 9 layers, and in some books it is written as 10 layers. So You can go with this, yeah, that is, you can say there are 10 layers in the retina. In this diagram, you can see the layers, retinal layers. So from outside to inside, this is the outer side, this is the inner side. That is, the light falls through, the light enters through this direction, and it falls on the pigment cells. Okay, this is the pigment cells. So this is the pigment epithelium, then photoreceptor layer containing the rods and cones. These are the rods, these are the cones. Then this is the outer limiting membrane. This is the outer nuclear layer, which consists the nucleus of rods and cones. Then outer plexiform layer. Okay. Then this is the inner nuclear layer consisting of the bipolar cells, amacrine cells. Then inner plexiform layer, all this fibers. Then the ganglion cell layer. Then we have got the nerve fiber layer, and finally the inner limiting membrane. Coming to the photoreceptors, that is the rods and cones. Rods and cones are the end organs of vision which transform light energy into visual nerve impulse. There are about 100 million rods and 3 million cones. The major functional segments of either a rod or cone are, and these are shown in this diagram. You can say this is the outer segment, this is the outer segment, then this is the inner segment. This is the outer segment and this is the inner segment. Then this is the nucleus and this is the synaptic body or the synaptic terminal. So this is the basic structure of, this is a rod and this is a cone. The main difference is the shape, this part, the outer part. In rod it is the shape of a rod, and in the cone it is conical in shape, so it is named as cone. The rest is same. You can see here, this is the plasma membrane. They are enfolded, foldings are there. Okay, and within this foldings there are the pigments. In the rod it is the rhodopsin, and in the cone these are known as simply the color pigments. In the outer segments of the rods and cones, there are a large number of discs I have mentioned, which are actually an enfolded shelf of cell membrane. There are as many as 1000 discs in each rod or cone. The light-sensitive photochemical rhodopsin and the color pigments are incorporated into the membranes of the discs in the form of transmembrane proteins. The inner segment of the rod or cone contains the cytoplasm with cytoplasmic organelles. Especially important are the mitochondria, which play the important role of providing energy. The synaptic body is the portion of the rod or cone that connects with subsequent neuronal cells, the horizontal and bipolar cells, which represent the next stages in the vision chain. Coming to the pigment layer of the retina, the black pigment melanin in the pigment layer prevents light reflection throughout the globe of the eyeball, which is extremely important for clear vision. This is the main function of the pigment layer. The pigment layer also stores large quantities of vitamin A, which is an important precursor of the photosensitive chemicals of the rods and cones. I suppose this was told to you the structural characteristics of fovea centralis. Foveal region has the highest visual acuity. What is visual acuity? Acuity of vision is the ability of eye to determine the precise shape and details of the object. So why this foveal region has the highest visual acuity? Mainly, and these are the three reasons. First, the density of cones in fovea is maximum. Foveal cones have long and thin outer segments. This shape allows high packing density. Rods are absent here. The second point, All other inner layers of the retina are pushed aside in this area. This arrangement allows light to reach the cones without having to pass through the inner layers of retina. So here it directly falls on the retina and thereby reduces distortion of the image. Then the third point, each foveal cone relates to single ganglion cell. We will see later on that the rods, they relate to two or more than one rod cell relate to a single ganglion cell. Okay, but in case of foveal cone cells, they one foveal cone relates to a single ganglion cell. So the nerve transmission, the impulse transmission is faster. That's why, for these reasons, the visual acuity is maximum in the fovea centralis. Now coming to the rhodopsin-retinal visual cycle. This is a very important topic. Rhodopsin emits decomposition and light energy. So rhodopsin is a combination of the carotenoid pigment retinal and the protein opsin. Rhodopsin is present in the rod cells, mainly in the outer segment of the rods. The retinal is a particular type called 11-cis retinal. The retinal which is found in rhodopsin, it is 11-cis retinal. When light energy is absorbed by rhodopsin, it decomposes within a very small fraction of a second to Bathorhodopsin. The cause of this rapid decomposition is photoactivation of electrons. Then this Bathorhodopsin is extremely unstable and decays in nanoseconds to lumirhodopsin. This product then decays in microseconds to metarhodopsin 1, then in about a millisecond to metarhodopsin 2. So we can see here, at first the rhodopsin, that is the 11-cis retinal of the rhodopsin. When light falls on the rhodopsin, the 11-cis retinal decomposes to Bathorhodopsin. Then this Bathorhodopsin decomposes to lumirhodopsin, lumirhodopsin to metarhodopsin 1, and metarhodopsin 1 decomposes to metarhodopsin 2. This metarhodopsin 2 decomposes much more slowly into the completely split products, all transretinal and opsin. Due to the separation of all transretinal from opsin, the reddish color imparted to opsin by the photopigment disappears and opsin becomes pale yellow. Hence the process is called bleaching. This process of decomposition of the rhodopsin is known as bleaching. It is the metarhodopsin 2, also called activator rhodopsin, that excites electrical changes in the rods, and the rods then transmit the visual image into the CNS in the form of optic nerve action potential. Now let's see how this rhodopsin is regenerated. The first stage in regeneration of rhodopsin is to reconvert the all trans retinal into 11-cis retinal, which is catalyzed by the enzyme retinal isomerase. Once the 11-cis retinal is formed, it automatically recombines with the scotopsin to form rhodopsin again. In the retina, under constant light stimulation, a steady state must exist under which the rate at which the photochemicals are bleached is equal to the rate at which they are regenerated. This equilibrium between the photodecomposition and regeneration of visual pigment is referred to as the visual cycle. I will show you the diagrams. So this is the whole decomposition of rhodopsin. Here you can see the rhodopsin. When light falls on the rhodopsin, it decomposes to Bathorhodopsin. Okay, this Bathorhodopsin again decomposes to lumirhodopsin within a nanosecond. Then this lumirhodopsin decomposes to metarhodopsin 1. This metarhodopsin 1 again converts into metarhodopsin 2. This metarhodopsin, it finally splits into scotopsin and the all-trans retinal. And this all-trans retinal, under the action of the isomerase enzyme, it can again get converted into 11-cis retinal. Okay, then this 11-cis retinal binds with scotopsin to form the rhodopsin again. So this cycle continues, and this is the visual cycle. Here another way in which the rhodopsin is regenerated, it is shown here. This all trans retinal, it is converted into all trans retinol. Okay, and this all trans retinol, it is a form of vitamin A. Then this all trans retinol, under the action of the enzyme isomerase, it gets converted into 11-cis retinol. And this 11-cis retinol, it again converts into 11-cis retinal. And this 11-cis retinal combines with scotopsin to form rhodopsin. So this is the visual cycle. Rhodopsin under light energy, it splits into all trans retinal and opsin. Then this all trans retinal, under the action of the enzyme retinal isomerase, it again gets converted into 11-cis retinal. Then this 11-cis retinal combines with opsin to form rhodopsin. So this cycle is the visual cycle. Role of vitamin A in formation of rhodopsin, I have told you. There is a second chemical route by which all trans retinal can be converted into 11-cis retinal, which is by conversion of the all trans retinal first into all trans retinol, which is one form of vitamin A. And this all trans retinol is converted into 11-cis retinol under the influence of the enzyme isomerase. Finally, the 11-cis retinol is converted to 11-cis retinal, which combines with scotopsin to form new rhodopsin. This is the pathway. All trans retinal, it gets converted into all trans retinol, which is a form of vitamin A. Then this all trans retinol, under the action of isomerase enzyme, it gets converted into 11-cis retinol. Then this again gets converted into 11-cis retinal. Then this 11-cis retinal joins with scotopsin to form rhodopsin. Vitamin A is present in the cytoplasm of the rods and the pigment layer of the retina. Is used to form new retinal when needed. Conversely, when there is excess retinal in the retina, it is converted back into vitamin A. Coming to the vitamin A deficiency. Vitamin A deficiency is due to inadequate intake of foods high in vitamin A or beta carotene. One of the earliest visual defects to appear with vitamin A deficiency is night blindness, which is also known as nyctalopia. Vitamin A deficiency also contributes to blindness by causing the eye to become very dry, which damages the cornea. This condition is known as xerophthalmia. And also, due to this dryness, the retina may be damaged. Then vitamin A first alters rod function, but concomitant cone degeneration occurs as vitamin deficiency develops. Prolonged deficiency is associated with anatomic changes in the rods and cones, followed by degeneration of the neural layers of the retina. So we can see vitamin A deficiency is really a serious deficiency, and which is mainly found in developing countries where there is malnutrition in the children. Coming to the phototransduction. So what is phototransduction? When light strikes the eye, a hyperpolarizing potential is generated in the rods and cones, which is known as the photoreceptor potential. The conversion of the light energy to neural signal by the visual pigments in the photoreceptors is known as phototransduction. Now let's see the mechanism of phototransduction. Light induces a conformational change in the photopigment and the associated G protein that alters membrane potential by lowering the cyclic GMP level. The change in membrane potential at the synaptic terminal modulates the discharge of the neurotransmitter glutamate and thus initiates a response in the next order of neuron. The mechanism of phototransduction can be better appreciated by understanding the processes taking place in the absence of light. So let's see what are the processes or changes occurring in the rod cell in the dark, that is when there is no light, when there is when there is no decomposition of the rhodopsin. Okay, so in the inner segment of the rod, the sodium potassium pump continually pumps sodium from inside the rod to the outside, and potassium ions are pumped to the inside of the cell. So this is a usual phenomena which is occurring. Okay, it also occurs in the inner segment of the rod. As in other cells, this creates a negative potential on the inside of the interior cell. So you see the sodium ion is pumped out of the cell and potassium ion is pumped inside the cell. So due to this, a negative potential is generated. However, in the outer segment of the rod, in the dark state, the membrane is leaky to sodium ion that flows through cyclic guanosine monophosphate cGMP gated channels. In the dark state, cyclic GMP levels are high. The cyclic GMP remains bound with the sodium channel protein of the rod's outer membrane in a way that splints it in the open state, permitting sodium ion to continually leak back to the inside of the rod and thereby neutralize much of the negativity on the inside of the entire cell. Thus, under normal dark conditions, there is reduced electronegativity inside the membrane of the rod, measuring about minus 40 millivolts. This decrease in membrane potential, in turn, keeps the voltage-gated calcium ions open in the synaptic terminal. The calcium-mediated exocytosis of synaptic vesicle results in a steady release of the neurotransmitter glutamate. So, this whole process will be clear in this diagram. So this is the rod cell, this is the outer, this is the inner segment, and this is the outer segment. In the inner segment, what happens? This is the sodium potassium ATPase pump. This pump continually pumps sodium ion from inside the cell, and potassium ions are pumped to the inside of the cell. Sodium ion out and potassium ion inside the cell. So this creates a negative potential on the inside of the entire cell. However, in the outer segment, in the dark state, the membrane is leaky to the sodium ion that flows through cyclic GMP gated channels. These are the cyclic GMP gated channels. So this cyclic GMP, this binds to the outer membrane with the sodium channel protein of the rod's outer membrane, and due to this binding, what happens, these channels, they remain open, and through these channels the sodium ions can leak inside the outer segment of the rod cell. So there is continuous influx of sodium ion through these cyclic GMP gated channels. So result, what happens? Under normal dark conditions, there is reduced electronegativity inside the membrane of the rod. So these are positive ions, and this will reduce the negativity of the whole cell inside of the negative, that is the negativity which was created due to the sodium potassium pump. This will be reduced by this continuous inflow of these positively ion sodium ions. Hence, the inside of the membrane, the electronegativity decreases and it measures about minus 40 millivolts. And this decrease in the membrane potential in turn keeps the voltage gated calcium channels open in the synaptic terminal. And this there is calcium mediated exocytosis of the synaptic vesicles, and as a result there is continuous release of the neurotransmitter glutamate. Now let's see what happens in the light. That is, when light falls on the rhodopsin, what happens? When the rhodopsin present in outer segment of the rod is exposed to light, there occurs photoactivation of the electrons in the retinal portion, and the rhodopsin starts decomposing. As I have already told you, the activated rhodopsin stimulates a G protein called transducin. The activated rhodopsin, that is the metarhodopsin 2, it stimulates a G protein called transducin, which activates an enzyme cyclic GMP phosphodiesterase. And this cyclic GMP phosphodiesterase catalyzes the breakdown of cyclic GMP to 5-GMP. Hydrolyzation of this cyclic GMP by phosphodiesterase removes the splinting and allows the sodium channels to close. And due to this flow of the sodium ion is blocked by the channel closer. So there is decrease of sodium flow inside the rod cell, and this excites the rod. So what happens in the light? This cyclic GMP gated channels are closed, and as a result the sodium influx stops in the light. Okay. In the inner segment, sodium ion continue to be pumped outward through the membrane of the inner segment. Thus, more sodium ion now leave the rod than leak back in. This creates increased negativity inside the membrane, and the greater the amount of light energy striking the rod, the greater the electronegativity becomes. That is, the greater is the degree of hyperpolarization. This hyperpolarization reduces the release of glutamate, and this generates a signal in the bipolar cells that ultimately leads to action potentials in ganglion cells. The action potentials are transmitted to the brain. So here, let's see what are the steps of this phototransduction. So when light falls on the rhodopsin, what happens? There is isomerization of 11-cis retinal to all trans retinal. And due to this change, there is conformational change in the opsin forming metarhodopsin 2. And as a result, there is activation of the G protein transducin. And this transducin activates another enzyme, which is the phosphodiesterase. And this phosphodiesterase converts the cyclic GMP to 5-GMP. So as a result, what happens? There is less amount of cytosolic cyclic GMP in the outer segment of the rod cell. So what happens? There is closure of the cyclic GMP gated sodium channels, and due to this closure of the sodium channels, what happens? There is decreased sodium influx causing hyperpolarization. Decreased release of, and due to this hyperpolarization, what happens? There is decreased release of synaptic transmitters, and due to this decreased release of synaptic transmitters, there is generation of response in the bipolar cells, and this uh, response is transmitted to the finally to the ganglionic cells, and from the ganglionic cells action potentials are generated which are transmitted to the brain. Now how this ends? Within about a second, Another enzyme, rhodopsin kinase, which is always present in the rod, inactivates the activated rhodopsin, the metarhodopsin 2, and the entire cascade reverses back to the normal state with open sodium channels. So this is how this cascade ends due to this enzyme which is known as the rhodopsin kinase. Till now I have described the photochemistry of rhodopsin. The photochemistry of coloration by the cones, it is also similar. The photochemicals in the cones have almost exactly the same chemical composition as that of the rhodopsin in the rods. The only difference is that the protein portions or the opsins called photopsins in the cones are slightly different from the scotopsin of the rods. The retinal portion of all the visual pigments is exactly the same in the cones as in the rods. So the whole photochemistry is similar in the color pigments that is in the cones. Now coming to the visual adaptation. So what is adaptation? Visual as adaptation. Visual adaptation refers to the ability of the retina to adjust its sensitivity according to ambient light. This ability allows the retina to operate efficiently over a wide range of lighting conditions, and it reflects the switching between the use of the cone and rod systems for bright and low light conditions respectively. So the retina, it can adapt itself to low light as well as to the high light, and this adaptation is known as the visual adaptation. Now let's see, visual adaptation may be light adaptation or dark adaptation. So what is light adaptation? When a person enters a bright lighted area from a dim lighted area, he feels discomfort due to the dazzling effect of the bright light. After some time, when the eyes become adapted to light, he sees the objects around him without any discomfort. This process in which the eye gets adapted to increased illumination is called light adaptation. So this is a common phenomenon which occurs with almost every one of us. So what is the basic mechanism of this light adaptation? This is because if a person stays in bright light for some time, large portions of the photochemicals in both the rods and the cones reduces to retinal and opsins. Purthermore, much of the retinal of both the rods and the cones gets converted into vitamin A. As a result, what happens? The photosensitive chemicals remaining in the rods and cones are considerably reduced, and the sensitivity of the eye to light is correspondingly reduced. Now let's see what is dark adaptation. Dark adaptation is the process by which the person is able to see the objects in dim light. If a person enters a dark room from a bright lighted area, he is unable to see for some time. So this is a common phenomena which occurs when we suddenly enter a movie hall. So after some time, after some time, our eyes gets adapted and we start seeing the object slowly. This process is known as the dark adaptation. This occurs because if a person remains in darkness for some time, the retinal and opsins in the rods and cones are converted back into light-sensitive pigments. Also, vitamin A is converted back into retinal to increase light-sensitive pigments. There is gradual increase in light sensitivity of the retina when in low light conditions. So due to this, there is dark adaptation, mainly due to the rod cells. Rods adapt to darkness slowly as their rhodopsin levels are restored, and indeed, it may take more than 30 minutes for the retina to become fully dark adapted. In contrast, cones adapt rapidly to darkness, but their adapted threshold is relatively high, and so they do not function when the ambient light level is low. Thus, within 10 minutes in a dark room, rod vision is more sensitive than cone vision and becomes the main system for seeing. In the dark adapted state, Primarily, rod vision is operative and thus visual acuity is low and colors are not distinguished. This is called scotopic vision. However, when the light levels are higher and cone function resumes, visual acuity and color vision are restored, and this is called photopic vision. There is an intermediate range of light levels at which rod and cones are both functional, and this is known as mesopic vision. What are the other mechanisms of light and dark adaptation? In adaptation, in addition to adaptation caused by changes in concentration of rhodopsin or color photochemicals, the eye has two other mechanisms for light and dark adaptation. The first is the change in pupillary size. This change can cause adaptation of approximately 30-fold within a fraction of a second because of changes in the amount of light allowed through the pupillary opening. The other mechanism is neural adaptation, involving the neurons in the successive stages of the visual chain in the retina itself and in the brain. So these are the two other mechanisms apart from the changes in the concentration of rhodopsin which help in other light and dark adaptation, that is in visual adaptation. The first change is the pupillary size. The pupillary size, it can constrict and dilate, and it can control the amount of light falling inside the retina. And the other mechanism is the neural adaptation, which involves the neurons in the successive stages of the visual chain. Now let's see the neural function of the retina. The different neuronal cell types of the retina are first the photoreceptors, the rods and cones which transmit signals to the outer plexiform layer where they synapse with bipolar cells and horizontal cells. The second is the horizontal cells which transmit signals horizontally in the outer plexiform layer from the rods and cones to bipolar cells. The output of the horizontal cells are always inhibitory. Therefore, this lateral connection provides the same phenomena of lateral inhibition, that is helping to ensure transmission of visual patterns with proper visual contrast. So due to these horizontal cells, we can see that is the image comes with a proper visual contrast due to this lateral inhibition. The bipolar cells transmit signals vertically from the rods, cones, and horizontal cells to the inner plexiform layer, where they synapse with ganglion cells and amacrine cells. Two types of bipolar cells provide opposing excitatory and inhibitory signals in the visual pathway. First, the depolarizing bipolar cell and the hyperpolarizing bipolar cell. That is, some bipolar cells depolarize when their rods and cones are excited and others hyperpolarize. The amacrine cells transmit signals directly from the bipolar cells to ganglion cells or horizontally within the inner plexiform layer from axons of the bipolar cells to dendrites of the ganglion cells or to other amacrine cells. The ganglion cells transmit output signals from the retina through the optic nerve into the brain. There are three distinct types of ganglion cells designated W, X, and Y cells. Each of these serves a different function. Finally, the interplexiform cells, though these are not very prominent cells, but it's believed that these cells transmit inhibitory signals in the retrograde direction from the inner plexiform layer to the outer plexiform layer, which control lateral spread of visual signals by the horizontal cells in the outer plexiform layer. Their role may be to help control the degree of contrast in the visual image. So this function is, the function of interplexiform cells is similar to the horizontal cells. Transmission of most signals occur in the retinal neurons by electrotonic conduction, not by action potentials. The only retinal neurons that always transmit visual signals by means of action potentials are the ganglion cells. Out of the different neural cells of the retina, only the ganglion cells, they send visual signals in the form of action potential. All the other neuronal cells, they conduct the impulse through electrotonic conduction. The importance of electrotonic conduction is that it allows graded conduction of signal strength. Thus, for the rods and cones, the strength of the hyperpolarizing output signal is directly related to the intensity of illumination. The receptor potential is approximately proportional to the logarithm of the light intensity. Okay, it is not like all and none phenomena. It is directly related to the intensity of the illumination. It is from the ganglion cells that the long fibers of the optic nerve lead into the brain. Because of the distance involved, the electrotonic method of conduction employed in the rods, cones, and bipolar cells within the retina is no longer appropriate. Therefore, ganglion cells transmit their signals by means of repetitive action potentials instead. Furthermore, even when unstimulated, they still transmit continuous impulses at rates varying between 5 and 40 per second. The visual signals in turn are superimposed onto this background ganglion cell firing. Finally, the processing of visual information in the retina. In a sense, the processing of visual information in the retina involves the formation of three images. The first image, formed by the action of light on the photoreceptors, is changed to a second image in the bipolar cells, and this in turn is converted to a third image in the ganglion cells. In the formation of the second image, the signal is altered by the horizontal cells, and in the formation of the third, it is altered by the amacrine cells. There is little change in the impulse pattern in the lateral geniculate bodies, so the third image reaches the occipital cortex. So all the later part of this visual information, how this is processed and how it is, this information is carried through the visual pathway, this will be discussed in the later classes. I suppose you have got a little bit idea regarding this photochemistry vision.
Transduction cascade. When light enters the eye, it travels past the ganglionic cell layer and the interneuron layers until it reaches the photoreceptor layer and the pigmented layer deep in the retina. In the photoreceptors, light is absorbed by rhodopsin proteins in rods and photopsin proteins in cones. Rhodopsin and photopsin are both G protein coupled receptors and are in the opsin family. G protein coupled receptors are cell surface receptors that receive a signal from outside of the cell and trigger changes within the cell. When the opsin proteins, rhodopsin and photopsin, are struck by photons, a protein called retinol changes shape or isomerizes from 11 cis retinol to 11 trans retinol, creating a conformational change in the opsin proteins. That causes the opsin proteins to activate G proteins called transducins, or GT, which in turn activate the enzyme phosphodiesterase. Phosphodiesterase converts cyclic guanosine monophosphate, or cGMP, to 5-guanosine monophosphate. Now, normally rods and cones have sodium channels which are bound to cGMP that allow sodium to continuously flow into the cell. When phosphodiesterase converts cyclic GMP to 5-guanosine monophosphate, there's less cyclic GMP within the cell available to bind sodium channels. Therefore, the sodium channels close. This decreases the inward current of sodium entering the cell, and the photoreceptor becomes more negative, or hyperpolarized. When photoreceptors are hyperpolarized, they stop sending action potentials down to the synaptic terminal, and that closes the voltage-gated calcium ion channels near the synaptic terminal. As a result, there's less of the excitatory neurotransmitter glutamate released into the synapse between the photoreceptor and bipolar cell. So, in essence, when light triggers a photoreceptor, it turns off and stops releasing neurotransmitters. Interestingly, it's the lack of glutamate that causes the bipolar cell to open up its voltage-gated calcium channels and depolarize. And the bipolar cell then triggers depolarization of the ganglion cell. And from there, the action potential is propagated along the optic nerve, and ultimately to the primary processing centers in the brain in the occipital lobe.